Okay, so welcome back. Let's take a look at the concept check questions, and then we'll move on to look at a couple of ways in which we can use DNA technology. So why can forensic scientists do a forensic analysis from as little as 15 cells worth of DNA? Because we can amplify it using the PCR technique. Remember, polymerase chain reaction allows us the opportunity to amplify small amounts of DNA to levels where we can comfortably do a forensic analysis. And PCR is now a very, very powerful tool as part of the toolkit for forensic DNA analysis. And what are STRs and why are they useful for DNA profiling? Remember, short tandem repeats are short sequences of nucleotides, usually around 4 to 6 nucleotides long, that are repeated multiple times back to back. And everybody has these STRs, and the length of these repeated sequences, the number of repeats in different locations, is a product of our inheritance. We get our STRs as part of our inherited chromosomes from our parents. And because each of our combinations of STRs is absolutely unique to ourselves, like our fingerprints, we can create a DNA profile if we look at enough of the STR regions, and typically we look at about 13 of them. We can create a unique series of STR profiles that looks very much like a genetic fingerprint, or acts very much like a genetic fingerprint. So that's why STRs are so useful in DNA profiling. So the next use of DNA technology falls under a heading called bioinformatics. The ability to make use of all of the huge amounts of DNA information that we have through DNA sequencing to start to understand how our genomes function. So there are two basic concepts that come out of the use of this information. What is known as genomics, which looks at the total function of all the genes that make up our genome, and then proteomics, which looks at the function of all the proteins that our cells are producing as a consequence of the genes. This is what is referred to as big data. And the reason why we can now take advantage of this big data is because of computer power. All of the genetic information, the sequencing information, the protein sequence information, these are stored in huge data banks. The NIH has a couple of data banks, one for genes and one for protein. The European Molecular Biology Consortium in Europe has another huge data bank that includes gene information, and in a subset also protein information. And when biologists generate new gene sequences or new protein sequences, they can enter this information into their desktop computer in their office and submit that up to the database, both to get it incorporated into the database, but also to use a search algorithm to compare their gene sequence against the known gene sequences or protein sequences. Because it turns out that genes and proteins exist as families. And there are genetic signatures and there are amino acid signatures that identify which genes or which families of genes and which families of proteins a particular gene sequence or a particular protein sequence might belong to. And because a lot of this information is public record in these databases, you can now learn a lot about a genetic sequence or a lot about an amino acid sequence without doing anything more than generating the sequence and submitting it to the data bank for comparison. This is the era of big data and big data analysis. So how do we get all of this information that goes into these data banks? Well, that's the product of DNA sequencing. As I said, very early on, sequencing was a very, very laborious task, but this is the reason why this picture illustrates the reason why it's no longer such a chore. Each one of these machines sitting on the bench here is a sequencing machine, and it's completely computer driven. All you need to do is to supply your DNA sequence material, your DNA material to be sequenced, and the machine does the rest. It not only cuts it up into smaller fragments, it runs technology that allows it to read the sequence of nucleotides from those fragments. It stores that information, and then it can run an algorithm that scans the nucleotide sequences generated by all of those fragments and can then splice all of those fragments together based on the overlaps of nucleotide sequences generated from each of the fragments. This is why it is so easy now to completely sequence a person's genome, about 3 billion nucleotides in less than 48 hours. Because you don't try and sequence the whole 3 billion nucleotides as a unit. You simply cut the DNA up into smaller fragments. Each fragment goes into one of these machines. Each of these machines only sequences the particular fragment or fragments that it's supplied with. All of that information is then entered into a computer that runs the algorithm that compares the sequences of all the fragments, looks for the overlaps in nucleotide sequences, splices the overlaps together to splice the fragments together, and in a short amount of time, several hours, it can splice together all the fragment information generated by all of these machines, several hundred of them generally in a sequencing laboratory, and generate a complete genomic sequence in less than 48 hours. And all of that can be done for at least for less than $200. These days, it's rapidly approaching less than $100. It's really, really, really easy to do this sort of thing now. And the sequencing of genes is becoming a very important tool biologically. One of the things that has always proved to be problematic in biology is understanding what is the relationship between different species and what is their evolutionary history. A lot of that could only be worked out or determined by doing actual comparisons between the organisms, looking at what is known as anatomy, internal appearance, and morphology, external appearance. And by and large, it turns out, based on our new information, based on DNA sequencing, the old anatomists and morphologists and taxonomists did a pretty good job of figuring out who was related to whom. But in some circumstances, big picture, and in some cases, small picture, relationships were not always as obvious as they might have been. But DNA has proved to be a really helpful tool. Because of course, if an organism is related to another organism through common ancestry, which is the whole basis of evolution, then they must also share some commonality at the DNA level. And so by comparing DNA sequences, we've been able to establish relationships between organisms. But because a lot of that sequencing had to be done by the old technology, by hand, in essence, only a few genes ever got sequenced in great detail across a number of organisms. But now with these sequencing machines and the relative ease of sequencing DNA material, a lot more information is now becoming available. And all of that information is becoming available through these public databases. That's where all of this information is winding up. In the NIH-based databases and in the EMBL, that's the consortium in Europe that does pretty much what the NIH does here, and their databases. There's also one in Japan now, if I recall rightly. I believe the Japanese equivalent of the NIH also maintains a database. But it is the use of these sequencing machines that's changed everything. Where did these machines come from? Well, these machines came from the drive to sequence the human genome. Now, the idea of sequencing the human genome first emerged back in the 1970s. People started talking about it. This was back in the day when we were sequencing 500 nucleotides in a month. You can imagine how long it would take to sequence the complete human genome containing some 3 billion nucleotides at 500 nucleotides a month. Even as the technology started to improve and we were able to start sequencing several thousand nucleotides a month, it was still considered, by the time the idea of sequencing the human genome actually started to become feasible in the late 1980s, as people started to think perhaps we need to invest money in doing this. And discussions between American researchers and European researchers and Japanese researchers about the feasibility of getting money from the various government authorities to support this sequencing were taking place, that perhaps it would take 10 years or more to completely sequence a human genome. As it would turn out, by the time they really got rolling on sequencing the human genome, it would take 3.5 years. And that was because several private entities got involved. Business set up by scientists from universities who resigned their university positions to start their own personal companies using investor money and dedicating their company to sequencing the human genome. And the person who really was at the forefront of all of this was a molecular biologist by the name of Craig Ventnor. And Ventnor set up his own company. It's still in business today and still doing very interesting research to basically sequence the human genome. And he very graciously offered his own genome as the genome to be sequenced by his own company. Yeah, right. But when he started this company and indicated that he was going to sequence the genome, people started to get really concerned, because if it was done by a private company, there was the possibility that that genome would then become proprietary information, and scientists would then have to pay for access to the genome sequence. The publicly funded concerns were going to make the sequence available to all comers for free, because it was paid for by tax money. So the government-funded teams and Ventnor's team sat down and discussed how to combine their efforts, and Ventnor eventually agreed to make all of the information that he would generate available through the public databases, and not to copyright all of this and patent all the information and start charging money for the sequence data. And as a consequence of this, a lot of effort was put into coming up with better ways of doing the sequencing. And this led to the first generation of sequencing machines. These machines in this picture are actually third generation machines. They are far faster and far nimbler at sequencing fragments than even the first generation machines were capable of doing. The first generation machines were what allowed the complete genome or the complete human genome to be sequenced in that 3.5 years. Now the third generation machines are sequencing a human genome in less than 48 hours. That's the level of progress that has been made. That means now that we have genetic information, genome information, not only for humans, but for lots of species. Some from the old hand sequencing, but many now through the sequencing, using the sequencing machines. And this is where genomics comes in. Because now we have a complete record of all the genes within a genome. Now we can start looking at how all of those genes are functioning in an organism. And that's where the microplate analysis or microplate arrays that we talked about in the last presentation come into play. Because there you can screen for the expression of thousands of genes on one little plate and see which genes are being expressed in different types of tissue. And that gives you a window. That's where the genomics information starts to come into play, because that gives you a window on what's going on in different types of tissue under different conditions. So here is a chart that shows the results of some of these sequencing projects, giving you an indication of the size of the genome. So it's talking about the haploid genome. Remember, that's one complete set of chromosomes versus the number of genes within the genome. So you can see that our genome is relatively large in terms of the number of base pairs, about 3 billion base pairs in a haploid set of chromosomes. And by the way, as far as the sequencing system is concerned, a haploid set of chromosomes is not 23, as you might suspect, but 24. And you might go, why? I thought humans had 46 chromosomes, 23 pairs of homologous chromosomes. So one set of chromosomes, a haploid number is 23. Where does 24 come from? It comes about because of the gender chromosomes. Remember, there are 2 gender chromosomes, an X and a Y. In females, there are 2 X chromosomes. So the full set of chromosomes for a female, the 23 chromosomes, includes 1 X chromosome. For a male, however, it's X and Y. So if you want to take into account a male's haploid number of chromosomes, if you only included the Y, you would be excluding his X. So in order to understand the full human genome, you have to sequence the 22 autosomes plus the X chromosome and the Y chromosome for a grand total of 24 chromosomes. So the human genome, in terms of sequencing the genome, involved 24 chromosomes, not 23, because we had to sequence both the X and the Y chromosome. Now, one of the things that is really interesting here is that the number of genes and the size of the genome has nothing to do with the complexity of the organism. So what do I mean by that? Well, look at the size of the corn genome. It's getting up to about 2.5 billion nucleotides. So it's getting up into the same range as ours. Yet, is a corn plant as complex as a human? Would you argue that? Would you argue for that? Would you argue against that? How would you argue around that? If you want another example, here's the number of genes in the simple little fruit fly. About 15,000 genes. Some people say 17,000 genes. In a human, it's around 21,000 genes. So there's not a whole lot of difference in the number of genes. But look at the size of the Drosophila genome in terms of base pairs. doesn't even make it to 250 million base pairs. But that's because there are only 4 chromosomes in a haploid set of chromosomes for the Drosophila. Well, actually 5, because you've got the X and the Y chromosome there as well. The gene density in the Drosophila is significantly higher than the gene density in a human. But the sheer number of genes is not that different, percentage-wise. Yet who's going to argue that a Drosophila is anywhere near as complex as a human? Complexity of an organism is not a function of the number of chromosomes, the size of the genome, or the number of genes involved. The complexity of the organism results from what we were talking about in the last presentation, the regulation of those genes, the utilization of those genes, and it comes down to the Hox genes. Maybe you remember that. If you don't, you might want to go back and take a look at last chapter's presentation and find the section on Hox genes to just remind yourself of that. It turns out that the fruit fly has a single set of Hox genes. Humans have 4 sets of the same Hox genes, whereas Drosophila have a limited number of ways of regulating the total number of genes that it's got, because it's got a limited number of those master control genes, the Hox genes. Humans can do a lot more because they've got a lot more of these master control genes, the Hox genes. And that's what leads to the complexity of a human. It's not the number of genes or the number of chromosomes, it's what you do with those genes and how you utilize them. And that's one of the things that this table reinforces. The number of genes, the size of the genome, which is a reflection of the number of chromosomes, has no correlation to the complexity of the organism. It's what you do with the genes that you possess that determines the complexity of the organism. We do far more with the number of genes that we have than a Drosophila does with the number of genes that it has. Hence, we are more complex. It is much simpler. So this is basically going through the general idea of what the process of sequencing is like. The shotgun method, where you simply take the DNA, cut it up into fragments, take those fragments, sequence them in the sequencing machines, feed all of that information into the computer lines up the fragments according to the overlaps and assembles a sequence on that basis. It's essentially what I told you. And this is what I had done to my genome. Remember I said at the beginning, I've had my genome sequenced twice, once by the National Geographic Society's project, which unfortunately is now finished, and once by 23andMe. Why choose to do it by 2? And the National Geographic was my first choice because it was contributing to scientific research, was simply because I wanted to compare the 2 results and see if there was significant difference between particularly the migration story that the 2 sequences told, because the National Geographic was focusing on human migration. The 23andMe sequence provided more information, but one of the things that it did focus on was the origin of my family as part of the migration of humans out of Africa. And basically, it didn't tell me anything that I really wasn't aware of. And the story that both sequences tell is pretty much the same. But 23andMe also provided some health information and some other information. And it does also make some contribution to ongoing scientific research. So I was more than happy to have that done. So here's another example of a sequencing lab. We don't need to show you more pictures of that, you've seen that. So here's the location of the database that's maintained here in the United States, known as GenBank, maintained by the National Institutes of Health, the NIH. The Human Genome Project, as I mentioned, We've talked about how that happened, or how that came about. The project actually ended, or the almost complete human genome was announced by President Bill Clinton in 1990. And this is the single biggest surprise that came out of that. When we first started looking at the human genome, we thought it was a blueprint, a literal blueprint that outlined in very great detail, gene by gene, every aspect of the human body. And therefore it was thought, well, maybe we contain 1 million or more genes. By the time we got to the stage of actually beginning the project, people were thinking, well, maybe several 100,000 genes. It was quite clear that 1 million to 1.5 million genes was way overestimate. But when the whole genome was fully sequenced and people were able to see the true number of genes contained in the human genome, this was the surprise. The first number quoted at the end of the project was no more than 25,000 genes. Then it was reduced to 23,000 genes. At this point, we're pretty confident that it's less than 23,000 genes, probably no more than 21,000 genes actually constitute the genes that make up a human. Again, it's not the number of genes that determine the complexity, it's what you do with those genes, and humans do a lot with those genes. And as I mentioned, the region outside of those regions that actually contain genes, previously known as junk DNA, we now recognize in some cases are derived from viruses, fortunately disabled. But remember, we have other things that are in there. We have the regulatory sequences. We've got all of those little RNA molecules that we were talking about in the previous presentation. The tRNAs and the ribosomal RNAs, they have to come from somewhere as a product of transcription. The microRNAs, the interfering RNAs, they all have to come from somewhere, and they lie outside of the actual genes that we know make up that 23,000 genes. But we still don't know what the vast majority of our DNA actually does. One thing that has been suggested, however, is the reason why such a large amount of quote unquote junk DNA exists in our chromosomes is because it increases the likelihood of crossing over. Right, you remember crossing over from a discussion of meiosis? It turns out that the likelihood of crossing over, swapping DNA between the non-sister chromosomes or chromatids, increases with the distance between the various regions of the chromosome. So if you've got two genes located really close together on a chromosome, it is very unlikely that crossing over will separate them by exchange of DNA. But if they're located at opposite ends of the chromosome, it is way more likely that crossing over is, in fact, almost certain that crossing over will swap them between the non-sister chromatids. And so by having very long chromosomes, by preserving this quote unquote junk DNA in the chromosomes, it simply increases the likelihood of crossing over, which of course increases the random genetic variation that we show from generation to generation. It may actually be one of the reasons why humans as a species have been very successful in the short amount of time that we've been present on the planet. We don't know yet. There's a lot more that we need to find out about our DNA, but we at least have now some tools that allow us to probe in there. That $1,000, I need to change that because it's way less than $1,000 now. None of my sequencing costs more than $200. In fact, the National Geographic, when I had it done, was $200. When I had it done by 23andMe, it was just over $100, and that was only a couple of years space in between the two events. So even in 2 years, the price came down quite dramatically. Some other things that we now know is that when Homo sapiens migrated out of Africa and first encountered Neanderthals, we mated. Why? Because you and I and every other individual who traces our genetic origin to that group of Homo sapiens that migrated out of Africa. So everyone who doesn't trace their origin to sub-Saharan Africa, which is most of us, which is very few of us actually. Most of us trace our origins to the group that migrated out of Africa. We contain a percentage of Neanderthal DNA. Somewhere between 2.1 and 2.5% of your DNA is derived from Neanderthals. Neanderthals and Homo sapiens had sex, because that's the only way Neanderthal DNA could have got into the human genome. So it's unlikely that the Neanderthals actually sort of faded away and disappeared because we outcompeted them. There may be a little bit of that to it, but certainly we had sex with them, we mated with them, because the evidence is quite clear. And this is because we've been able to extract DNA from Neanderthal bones and apply modern DNA technology and splice together the complete Neanderthal genome. And it is very, very similar to our own. It is remarkably similar to our own. It may very well be that Neanderthals had language. They certainly had culture. They contain the same genes that we believe are associated with our ability to talk. So it's quite possible that they had language and that they had very complex societies, maybe even as complex as ours. One of the things that we do know is that their brains were a little bit larger than ours, about 1.2 times the volume of our brains. So if you are a believer in the size of the brain is a measure of intelligence, which by the way is not the case, the size of the brain is not a measure of intelligence, or the volume of the brain is not a measure of intelligence. But in the past, that has been an idea that has been proposed. Then you would have to argue that Neanderthals may have been more intelligent than we are. And yet our image of Neanderthals is that they were primitive grunting cavemen. That was not the case. They may have been every bit as intelligent as we are. They may have had the same culture that we had, or a similar sort of culture. They had some form of religious belief. We know they cared for their sick and aged. We know that they buried items of importance with their dead, and we know they buried their dead. We know that they had the genes that would have permitted them to have language, because they're the same genes that we possess that we firmly believe are associated with our ability to have language. And we had sex with them when we encountered them, or when our ancestors encountered them. And there's even now evidence that suggests that Neanderthal genes were actually carried into sub-Saharan Africa. Not only did Homo sapiens migrate out of Africa, but after having met the Neanderthals and Neanderthal DNA being incorporated into the Homo sapien genome, that some Homo sapiens returned back into Africa and took Neanderthal genes with them, because there is now evidence of Neanderthal genes in some regions of sub-Saharan Africa. So either Neanderthals themselves migrated back in, or Homo sapiens that carried Neanderthal genes took them back. Because the evidence suggests that this took place around 6,000 years ago. Lots of information we can learn by looking at genomic sequences. So, just out in case you're interested, my genome contains about 2.2% Neanderthal DNA, it's a little bit on the lower side of average, is what the two reports that I have indicated. They said that my level of Neanderthal DNA is a little bit on the low side of average, whatever that means. We also know that some of the conditions that we recognize in Homo sapiens as being adaptive for Homo sapiens, like pale skin in northern climates, that may have actually been something that we inherited from Neanderthals. Skin color, hair color, and things like that may be a product of some of the genes that we inherited from Neanderthals, and allowed us to adapt into environments where there was low levels of sun, and allowed us to absorb the ultraviolet radiation to produce the vitamin D that we need to maintain healthy bones in northern latitudes where there's low sunlight level, because Neanderthals were already adapted to that. Unfortunately, we also seem to have inherited some genes for genetic disorders that are associated with a type of cancer, and also perhaps with heart disease. We may have inherited the genes for those conditions from Neanderthals as well. Whether they were problems for Neanderthals, we don't know, but certainly those genes seem to be of Neanderthal origin, and they are problematic for us. We also have the ability now to compare sequences with closely related species like the chimpanzee, our closest related living relative. And our DNA is about 96 to 98% identical. In fact, as best we can tell, we both share essentially the same set of genes. So why the species difference? Well, we know that there are 6 distinct regions of difference between Homo sapiens and chimpanzees. And those 6 distinct differences lie not in the structural genes, but in the regulatory genes, the Hox genes. And it appears that that may be the driving force behind the divergence of Homo sapiens from the shared ancestor that we have with chimpanzees, and the reason why our two species went in very different directions evolutionarily. Because changes occurred in the regulatory genes, the Hox genes, in humans that did not occur in the chimpanzees. And of course, as we talked about in the last presentation, a mutation in a Hox gene has huge knock-on effects because they control hundreds of other genes. And so just changing the expression of a single Hox gene changes the expression of hundreds and hundreds of genes downstream from that. And so six changes may have been what created the huge difference between ourselves and chimpanzees and the direction that we both species moved in evolutionarily. We can also use genetic information to trace the origin of genetic diseases. So we've talked about a couple of these in other presentations. So I talked to you a little bit about the origin of cystic fibrosis as a response to the presence of cholera. We think that the mutation for cystic fibrosis actually evolved in the Indus River Valley, where cholera is known to have originated. The cholera bacterium is known to have originated, and the cystic fibrosis allele provides protection against the dehydration that's associated with cholera infections. So it may have been an advantage at that point. The same with the sickle cell allele in malaria zones provides a benefit if you're heterozygous, because it protects you against malaria, but you don't suffer the ravages of sickle cell. So here's another disease that I had never heard of until I read this section in the textbook. So Behçet's or Behçet's disease affects blood vessels, and it is found along the Silk Road. And this is the Silk Road leading from the Middle East through to China. This is the road, in fact, that Marco Polo followed when he entered into China and came back on again when he came back out of China. And there has been trade backwards and forwards along this road for the best part of at least 3,000, maybe 4,000 years, maybe even longer than that, because it's suspected that there has been trade between China and the early dynasties of Egypt, which goes back at least 3,000 to 3,000 BC or BCE, depending on which terminology you want to use. But so at least 5,000 years ago. And there may have been, there is evidence of trade between China and ancient Egypt. So the Silk Road has been active for a long time, and that also provides for a great opportunity for genetic disorders that evolve in one location to be passed from population to population along the Silk Road as individuals move along the road, either traveling the full length of it or traveling part of it and carrying their alleles with them. So there is a study reported in the chapter about this little boy named Nick Volker, who had a genetic disorder of his digestive system, and it meant that the only way to feed him was to do so through liquids, because his digestive tract simply could not tolerate solid food and could not absorb food as a product of digestion. And doctors struggled for a long time to try and figure out what his problem was. Eventually, having basically made a lot of mistakes, they started to think in terms of, well, perhaps this is a genetic disorder that no one has discovered before. And maybe what we really need to do is take a look at the genetics of his immune system, because it's an autoimmune disease, and find out if he, in fact, is carrying a very rare mutation that has never been described before. And so they compared the genetic sequence of a very highly conserved part of the immune system genetics, and discovered that indeed Nick had a mutation in a particular amino acid, and you can see it highlighted here, that did not exist anywhere else. In fact, the particular amino acid that had been changed in Nick's situation was very, very highly conserved across all of the organisms that were investigated. In other words, this particular amino acid was absolutely critical to the correct functioning of this protein. And the fact that Nick had a change in that amino acid, and that it was just that amino acid that of all the amino acid changes that he possessed was the one that was so highly conserved on it in every other example of the protein that was looked at, that was significant. This must be the cause of all of his issues. Once that was recognized, and they realized that this was the situation, then they could go ahead and identify which of the genes was actually responsible. And it's this gene called the XIAP gene. And he has a single nucleotide mutation in that XIAP gene that gives rise to that single amino acid change. What that allowed the doctors to do was then target his bone marrow using gene therapy and replace his bone marrow with donor cells from a donor that had fully functional XIAP genes. This is the alternative way of fixing the bone marrow. If you are unable to actually fix the genes, you simply destroy the bone marrow. You do that with radiation, and then you inject a small amount of bone marrow taken from a donor, and that bone marrow then propagates to replace the now destroyed bone marrow. And in this case, the XIAP gene was fully functional, problem solved. This technique has actually been used in a number of situations. About 10 years ago, I was talking about sickle cell as a condition, and a young man in the class put his hand up, and I may have told you this story, I'm not sure. The young man in the class put his hand up and said, "I'm actually the first person to have their bone marrow replaced as a treatment for sickle cell." And he had had it done at St. Jude's Hospital in where is St. Jude's? It's, I want to say Alabama, but I may be wrong. But anyway, he had it done at St. Jude's, and he was able to bring in the little newspaper that St. Jude's publishes from the time when he was there, and it had a picture of himself and his doctor on the front and a little article inside talking about this treatment, and it had cured his sickle cell. It hadn't replaced his genes, it had just simply replaced his bone marrow. The young man actually came back and took Bio 2 with me a couple of years ago. And it was really good to see him again because he is doing quite well. His transplant, which was done when he was 7, took, and he responded very well to that. And he has essentially been living a normal, healthy life ever since. And he no longer suffers from sickle cell because his bone marrow produces normal red blood cells. So this is a very common alternative technique, but the gene therapy technology that we talked about earlier is the one that people are focusing on now, because that allows you to keep the original bone marrow in place. And bone marrow transplant is a tissue transplant, which means that you now have to take anti-rejection drugs, which weakens your immune system a little bit, which makes you more susceptible to infectious diseases and so forth. You are immune compromised is the situation that we talk about there. But being able to retain the original bone marrow and simply alter the genetics of the cells in the bone marrow means that you don't have to confront all of that, because all you're doing is fixing the genetic problem. You're not actually replacing the bone marrow. So gene therapy holds out a great deal of promise if it can be made to work reliably. And so we can talk about using it to look at various types of diseases and use it again forensically. In this case, to identify a dentist who had deliberately infected his patients with HIV, knowing that he was HIV positive. That West Nile virus came from birds and infects people via mosquitoes. That anthrax that was mailed to politicians in Washington and also was mailed to a book publisher in Boca Raton, all originated as a specimen from the Army's Biological Warfare Research Institute in Maryland. And they even narrowed it down to one individual who had access to the anthrax that was identified as the source of the problem, but they could never demonstrate it that was in fact this individual, and he constantly denied it. And eventually he committed suicide as a response to the constant pressure of being accused of being the person of having done this, even though that they could never prove it. They could just they kept the pressure up, trying to get him to admit that he was the one who did it, and he kept denying it, and eventually I guess the pressure got too much and he eventually wound up committing suicide as a consequence of this. But they were able to demonstrate that all of the anthrax that wound up in this book publisher, and unfortunately 3 people died as a consequence of the infection, and the anthrax that was mailed to Washington, to people in Washington, politicians in Washington DC, and also members of the media, all originated from the same source, and that one source was then traced back to Fort Detrick. So using genetic information is a very, very powerful tool in a number of different ways. I've talked about a lot of different ways, so I'm just going to keep going through these slides. If you want to talk about, if you want to read about some of those, you can read those slides. You can download the presentation and read those slides. Then we have this idea of proteomics, where rather than focusing on the genome, we focus on the proteome. So the genome is the set of genes that an organism possesses. The proteome is the set of proteins that are the product of those genes. So genomics looks at the action of all of those genes working together. Proteomics looks at the function of all of those proteins working together. So this is big data analysis again. Proteomics is relatively new, but it is springboarding off the use of AIs, artificial intelligence, and huge amounts of computing power to investigate genomes, to also investigate proteomes. And this has led to this concept called systems biology, where rather than looking at individual genes, we now look at whole systems, whole organisms. And in fact, a new way of identifying new organisms, and particularly bacteria and archaea, is not to identify the individual organisms, but to identify the presence of their unique genetic signatures. So there are databases of the genomes of all the known bacteria and all the known archaea. And so when people take specimens from new environments and they extract the DNA material and start sequencing through that, they can compare the derived sequences against the databases and rule out all the known organisms. So when they find a new sequence, they know that there is a new bacterium or a new archaea in that environment. Then they know what to start looking for. But their first identification of the presence of a new species is on the basis of the DNA signature, not on the basis of actually discovering the organism itself. So there are a number of different ways of looking at this. We talked about personalized cancer treatment. This is another example of systems biology at work, where you take a look at an individual's cancer and you take a look at the unique set of mutations that have led to that particular cancer in that particular individual, and you base your treatment on those sequences, on those mutations, because different types of cancer respond to different types of drugs on the basis of what genes have been mutated, and there's no point in using a drug that's not effective against the particular combination of mutations that have caused that cancer. Much better to identify the mutations that have caused the cancer and then pick the drugs that target those particular sets of mutations, because they're likely to have the greatest effect. So this is where the origin of personalized medicine is emerging from, out of this systems biology approach. Okay, one more break and then a few more slides and we're done. This is a really long chapter, I realize, and there's no real way around this. It's just got so much information in it. And as I said back at the beginning, there are so many cool things that we could talk about, and I'm not even scratching the surface of a lot of this. So here are our concept check questions. Approximately how many nucleotides and genes are found in the complete human genome? And I'm not even going to stop for a break here. I'm just going to answer these questions. So the human genome, it's 3 billion nucleotides and about 21,000 genes. What is the difference between genomics and proteomics or proteomics? So genomics, we just talked about this, genomics focuses on the genome and the functions of the genes as a system. Proteomics focuses on the proteins, or the proteome, we should say, if we're going to use the same terminology, and the functions of the proteins as a system. So that would be the difference between the two. Okay, let's finish off the last few slides so we can finish up this presentation. So there are a number of ethical and regulatory concerns that we need to also add to the mix here, because as soon as you start altering the DNA using DNA technology and you start using bacteria and viruses as vectors, there's always the potential for creating something that could escape from the laboratory and cause a problem. In fact, this is one of the concerns that people have raised about the COVID virus. Is this in fact a naturally occurring virus, or is it the product of lab science gone wrong? I don't know what the answer is to that. I'll wait for the experts to do their analysis. In terms of what the experts have said so far, on the basis of what we know about the genetic sequence of the COVID virus, which is well known, it actually appears to be a naturally occurring virus, not the product of genetic engineering. Some people are not willing to accept that and are saying that it is a lab-derived virus. I don't know what the answer is. And I think part of the problem is that people have become so skeptical about the nature of science over the last few years that even if scientists were able to present convincing evidence that demonstrated that it was a naturally occurring virus, or that it was the product of a lab, whichever way the evidence seems to point, there are always going to be people who are going to say, I don't believe you. And that's a problem. Because speaking as a scientist, I am more inclined to accept the data that I am presented with than someone's personal opinion. Because the data supports a concept. A personal opinion is just that, a personal opinion. That's where I stand on it anyway. So I'll wait for the data to come in and see what the evidence suggests. So various governments around the world have instituted various sets of regulations to control the research. There are various levels of biohazard laboratories, ranging from biohazard level 1, which is a fairly simple biohazard and very few precautions need to be taken other than standard sterile techniques and and so forth, all the way up to a biohazard level 5, where you have to wear a full hazmat suit. It looks a little bit like a simple spacesuit. You're completely enclosed in it with a separate air supply. The labs have what is referred to as negative pressure, meaning that the air is constantly flowing in and doesn't flow out, except through a series of sterilizers that include very fine filters, sterilizing equipment in the form of ultraviolet and heat to kill any possible organism that could be airborne. And that all takes place under multiple situations before the air is allowed to be returned to the environment. It's very, very expensive laboratory space to build. And there are very few of those level 5 labs around the world. One of them happens to be located at Fort Detrick. The Army maintains it. And that is where the anthrax bacteria came from. But it's because that is a level 5 containment laboratory that anthrax is actually found there, because that is the degree to which you have to maintain the security of anthrax. It is such a deadly disease when it gets into humans. In the case of genetically modified foods, in the United States, as I mentioned earlier, there is no requirement to label foods as containing genetically modified organisms. But we know that in the case of those foods that I mentioned earlier, the vast majority of those crops here in the United States and in a number of other countries, they are genetically modified, and it is predominantly genetically modified versions of those crops that are grown. Where you see the GMO label on foods, it is typically to indicate that the foods are non-GMO. That's strictly voluntary. There's no requirement for that label either. But some manufacturers feel that it is important to let customers know that there is no GMO derived material in the food. Personally, I have no particular opinion one way or the other on eating genetically modified crops. Considering that we have been modifying plants for many thousands of years now, and all of those genetically modified plants are artificial in some form or another. They're not as they were originally found in nature, and we have not suffered from eating those modified plants. And given what is the nature of what happens to the gene products when we eat those foods, they're all broken down via digestion into their component subunits. So no, we don't have the risk of incorporating a foreign gene into our body as a product of eating genetically modified foods, because those genes get broken down into their component nucleotides long before they're taken up in our digestive tract. The same with the proteins and any other products that are other macromolecules that are produced in genetically modified foods. So in terms of eating the foods, I have no particular problem in eating them. But where there may be an issue in genetically modified foods, and particularly in genetically modified plants, there is a possibility of the escape of those genes into wild close relatives of those crops. Because remember, most of our crop plants are derived from plants that grow wild. They've been modified by selective breeding, but they are still derived from plants that grow in the wild. And many of the wild ancestors are still growing in some locations. So if pollen from one of these genetically modified plants was to escape, it could potentially pollinate one of its wild relatives and take whatever gene modification has been made to the plant to that wild relative. So if it was a herbicide resistance gene or an insecticide gene or something like that, the wild relative could inherit that genetic component from its domestic relative. At the moment, that doesn't seem to be an issue. I know that in the case of the companies that have been creating these genetically modified plants, their requirement is that all of the gene products, or all of the seed products of those plants be utilized as food, that you cannot replant those crops. This is one of the arguments against genetically modified plants, by the way, that the farmers are forced to buy new seed every year. And in economically disadvantaged countries where these crops are a huge advantage because of the increase in output that they produce, traditionally the farmers would simply keep back some of the seed, like corn seed, wheat seed and so forth, and reuse that as the seed for planting the next crop. With transgenic plants, the farmers are not allowed to do this. It is part of the requirement that if they use this seed, they are not allowed to keep back some of the seed for growing the next year's crop. They have to utilize all of that. And so that's one argument against it. And the other, this idea of the genes being able to escape into the wild. I know the companies are very careful about engineering the plants so that if this should happen, the pollen would be incompatible with their wild relatives, that the pollen can only fertilize the plants that contain the genetic modification. There are ways of creating pollen incompatibility or pollen compatibility. It's actually a way in which plants that can self-fertilize are able to first self-fertilize because the pollen is compatible with the flower that it comes from, versus plants that cannot self-fertilize because the pollen is incompatible with the flower that it came from. It has to actually go and pollinate a flower from another plant. And so it's possible to genetically engineer pollen incompatibility into the pollen so that it can't pollinate its close relatives. And that seems to have worked so far. But there's always the possibility of something going wrong. And that's the reason why there are all of these protocols in place. Some of them are country specific, and in other cases, these are agreed upon internationally and are applied in all countries, but it's these international protocols that also allow for the import and export of genetically modified organisms as food shipments from countries in other locations. And if you feel uncomfortable about that, think about that next time you go to the supermarket and you find foods, fruits and vegetables in the supermarket as fresh that are not available locally because it's the wrong time of year. The only way that you can get those fruits and vegetables in the supermarket all year round is because they're being grown in other countries and exported to America to be made available in the supermarkets. So that's something to think about. Again, I'm not saying that that's necessarily correct or incorrect. That's just what is happening. So if you feel uncomfortable about importing plants of unknown origin from countries outside of the United States, then don't buy those fruits and vegetables when they're out of season locally, because you know where they came from. They must have come from outside the United States. It's a decision that you have to make, and it's a very personal decision. There are a number of agencies here in the United States that regulate genetic engineering. So you've got the FDA, you've got the EPA, you've got the NIH, and the USDA. These are the agencies that are responsible for regulating genetic engineering research and giving approval for the sale of genetically modified organisms as part of the food chain. In the case of human genetic engineering, yes, we can cure genetic diseases, but do we want to take it one step further and actually manipulate our genome to affect future generations? Not just to affect the individual who's got the genetic defect so that they can have a normal, healthy life, but to also affect future generations that may be produced by that individual. Is that a step that we want to take? Because that is the next step in the use of DNA technology in humans. Right now, all the changes that we make are only for the individual that has the genetic defect. There is no attempt to fix that defect so that it will not be passed on to the next generation. So whether it be a bone marrow replacement, like my student, he can still pass on the sickle cell allele to his kids because it's still present in his germ cells. It was only the bone marrow that was changed. Or if we do human gene therapy on bone marrow cells, that still only affects the current individual because the change is not being made at the gamete level, at the germ cell level in the reproductive organs. But what if you go ahead and make genetic changes that do affect the reproductive organs and therefore the gametes? That change will then be propagated to the next generation and subsequent generations. Are we willing to do that yet? That may very well be a question that your generation will be asked. In the meantime, because of the availability of genetic profiling technology, a number of legal issues have been raised. Like, is it legal to collect DNA from people who are arrested? And the Supreme Court has said, yes, it's just like taking a fingerprint and a photograph. So taking DNA, if you get arrested and DNA is taken to create a DNA profile, just consider it as being part of the arrest procedure. Because as far as the court is concerned, it's the same as taking a fingerprint and taking a photograph. In terms of your personal DNA profile, if you choose to have your genome sequenced, this becomes a really tricky situation. This is one of the reasons why I chose to go with 23andMe and not Ancestry.com. You may recall that I said that police forces have used DNA profiles to identify relatives of people that they suspected had committed crimes on the basis of public databases. Well, these public databases are databases like Ancestry.com. And Ancestry.com particularly has been designated as a database that police forces have used using DNA profiles to find relatives in Ancestry.com to be able to then make a case against the individual that they suspected of being the perpetrator of the crime on the basis of familial patterns. In the case of getting sequencing done for health reasons, this is where you really have to be careful because, again, the Supreme Court has indicated that the person who pays for your health insurance is the person that actually owns, or the entity that actually owns, your health records. So if your health insurance is supplied by your employer and paid for by your employer actually owns your medical records. You don't, even though they're yours personally. So if you were to get a gene sequence analysis done using your health records, that information is now available to your employer. And the potential for what they can do with that information is an open book, an open door. Now, the courts have already decided in a couple of situations things that they can't do. So they currently can't deny you health insurance on the basis of genetic information. Because again, possession of a mutation that is associated with a genetic condition does not automatically mean that you're going to have that genetic condition. Likewise, other types of insurance cannot be denied as a consequence of availability of genetic information. But some individuals have said, and some legal authorities have said, that under the circumstances, it might be better to have those sorts of analyses done at your own expense, because then you really do own the results and they don't turn up in your health records. So, as much as I was willing to have my genome sequenced by the National Geographic, I was comfortable that they would maintain the privacy of my material. And by 23andMe, which to the best of my knowledge has not been approached by the legal authorities yet to provide genetic information. The fact that my genome sequence is publicly available, if the right people ask with the right legal authority, is one of the prices that I have to pay for having had my genome completely sequenced. I'll live with that. Because if I should be unlucky enough to develop a genetic condition like cancer that requires access to my genome, well, that information is already available, and I can request that information be available to my physicians. But again, this is a very personal decision that all of us in the future are actually going to have to be required to make at some point in the future. I'm very confident of this. So here's some of the legal information that relates to genome sequencing. So you can see here that you cannot be denied health insurance on the basis of genetic information. You cannot be denied employment on the basis of genetic information, which would be the other big issue related to health insurance provided by your employer. Other interesting things associated with human genome sequencing, and we'll finish up on this one. It turns out that Genghis Khan, or Chinggis Khan, as the Mongols would prefer him to be called, was a very prolific individual. He basically took the philosophy that when he conquered a tribe or a people, he would take one of the daughters of the ruler as a member of his harem. And apparently he liked to sleep around with all the members of his harem, and he frequently fathered sons, children, and a lot of sons through this. And of course, the Y chromosome is the chromosome that gets passed from father to son. And it turns out that about 8% of the males living in Central Asia carry the Y chromosome originating from Chinggis Khan as a consequence of all of this. If you look at the men in Ireland, again, about 10%, 1 in 10 Irish men carry a Y chromosome from this ruler, Niall of the Nine Hostages, who was a ruler in Ireland in the 1400s. Apparently he was quite prolific too. And then this is an interesting one. There is a group of people living in southern Africa who have always practiced Jewish traditions, and their folklore says that they are descendants from a group of Jews that migrated down into Africa, and they considered themselves to be of Jewish origin, even though they live in southern Africa. And genetic analysis of the Y chromosome has indicated that yes, in fact, they are of Jewish origin. They really are Jews. And as a consequence, They have been given the right to migrate to Israel. They have been welcomed to Israel, and a large number of them have actually migrated to Israel because they want to live in Israel as the Jewish homeland. There are a number of other peoples around the world that have claimed connection to Jews, particularly along the Silk Road, because it is known that, or it is, again, folklore, that a number of Jews have traveled along the Silk Road, including several of the disciples of Jesus were rumored to have migrated or have traveled the Silk Road as far as China. So it's possible that if people were to do a similar sort of analysis, that they may discover small populations of individuals who are related to Jews as a consequence. All of this can be determined by looking at DNA information. So there are a lot of interesting things that we can learn from DNA sequencing, a lot of concerns, legal concerns, ethical concerns that we still need to sort out. As I said, I think these are going to be decisions that all of us are going to have to make. I've had to make a few already as far as my personal situation is concerned, but that was my choice. But I suspect that many of you are going to have to, at some point, make decisions about some of these questions, because I don't think DNA technology is going to become a lesser part of what we do. I'm pretty confident that the use of DNA technology is going to become a greater part of what we do as humans. Anyway, that brings us to the end of this chapter, and of course to the end of the semester as far as the presentations are concerned. One more concept check questions. Yes, you knew that there were going to be a couple more concept check questions. So what is the main concern about adding herbicide resistance genes to agriculturally significant plants? That those genes may escape into their wild relatives as a part of cross-pollination from the agricultural plants into their wild relatives. That's the big concern. The engineering, the genetic engineering is supposed to have created a situation where that is impossible, right? And so far that's been successful, but it remains to be seen if that is going to be a consistent thing. Why does genetically modifying a human gamete raise a different ethical question than genetically modifying a human somatic cell? This comes down to, does it affect just the individual, or does it affect subsequent generations? If you modify a somatic cell, then the effect is only on the individual in which the genetic modification occurs. However, if you modify a gamete, you're not just affecting the current individual, you're affecting subsequent generations, because now that genetic modification is going to pass into subsequent generations, or potentially be passed into subsequent generations. And that has greater implications than just affecting the life of a single individual. And that's where a huge part of the genetic and, the ethical and legal concerns over genetically engineering humans starts to emerge. Anyway, that's it for the semester. We have one more exam, the final exam next week. Check the syllabus to find out what day that is to be held. It is comprehensive, so it is covering the whole of the semester, but there will be questions covering these last two chapters, 11 and 12, on that exam, along with questions that cover the remaining chapters, chapters 1 through 10. So all 12 chapters will be covered on the final exam. If you, again, as always, if you have any comments, concerns, or questions about anything that I have covered in this presentation or any of the other presentations throughout the semester, by all means, come and see me during office hours. I'll be more than happy to go over anything that has been an issue for you. So at this point, I want to wish you good luck on the final exam. It's been an interesting experience teaching this class, considering that I redesigned the whole course concept before we began this summer. I've enjoyed it. I know that some of these presentations have been incredibly long, but I hope that breaking them up into smaller chunks and encouraging you to take breaks along the way has helped you maintain a level of interest and a level of focus that has allowed you to take in the material. I hope you found it interesting and thought-provoking and challenging, and that it serves you some purpose and benefit into the future. I'll see you around. Bye.